Thanks, Tim, and thanks, Bruce, and the Society for Translational Oncology and the MGH for allowing me to come back home and participate in this session. It's always fun as well to follow one of your residency colleagues as part of a session, and it's really been a great session already with two fantastic talks. And I'll try to, I think, pick up on some of the concepts that the last two speakers brought, at least, to try to give us a cohesive theme as we talk about myeloid malignancies. I'll just point out that I've had consulting and advisory roles for both Novartis and Agios, but I won't discuss any investigational uses of any drugs. So what I'd like to do in the next 35 minutes or so is bring us back from myelodysplasia to AML. And I was hoping to start this talk on a positive note and tell you that we've made stunning progress in AML compared to solid tumors. But this slide from MD Anderson, where they looked at all of the patients treated at MD Anderson, the majority of them on clinical trials with AML, over the past 4 decades would say the exact opposite. And this would suggest, despite the fact that we have effective chemotherapeutic agents for now 4 decades, that the overall survival of AML, although you can get a p-value that makes a statistician happy, does not result in a survival curve that makes us nor our patients happy. And I think for the standpoint of those of us who think about AML as a disease, both biologically, therapeutically, and clinically, this is the gauntlet. The fundamental question in this disease is we've made very little impact in the survival of AML, with the exception of acute promyelocytic leukemia. And the question fundamentally is why? And the posit that, or straw man that I'll use is that I think it's an inadequate historical understanding of the biology, and it's only with some of the newer insights that the other speakers brought up and I'll try to resonate with, that we're beginning to now have all the tools of the trade that will allow us to hopefully develop therapies that will bend this curve significantly so that we can be proud of the impact we make in AML in the next few years. And as this slide emanates, and this is the same show that I think that Tim showed in his slide from the Cancer Genome Atlas in AML, we've taken a very specific view of AML and asked, are there specific types of mutations that are of value from a biologic standpoint that might have therapeutic relevance? And it wasn't lost on us or anybody else that a substantial fraction of patients with AML have mutations either in genes that affect DNA methylation or in genes that affect chromatin modifications. And that had important biologic implications because that meant that these were new classes of genes that are important not only in AML, but we now know in all human cancer, but also the idea that the manipulation of the epigenome is important to AML suggested an inherent reversibility. Maybe the idea that some of the effects of these mutations are not indelible and rather can be therapeutically reversed. And I'll come back to that towards the latter half of my talk. And coupled with those discovery studies, we have prognostic studies in AML that our group and others have done, similar to the work that Ben presented in MDS, that have shown that patients who have mutations in known or putative epigenetic proteins, such as TET, ASXL1, DNMT3A, or MLL, do very poorly. This is a ECOG trial where patients were treated with dose-intensified daunorubicin and Ara-C and were offered allogeneic transplant if they had high risk. So this is the best therapy we have to offer, and these patients do very, very poorly. They have low response rates to induction therapy and a high relapse rate even if they do go into remission. So it's this subset of AML patients, we believe, that's driving a lot of the bad players in this disease and is very different, for example, than core binding factor leukemia, which is highly chemosensitive and often cured even without an allogeneic transplant. And so the question that our laboratory and many others have tried to address in the last few years is, if patients with mutations in epigenetic regulators that either affect DNA methylation or affect histone state, and you can see effects on histone tails here, and effects on DNA methylation, if these are bad actors, we need to understand the molecular circuitry that governs how they contribute to transformation. And then if we can do that, we need to think about therapeutic approaches to see if we can reverse these fundamental biochemical switches and see if we can actually restore leukemic cells to their normal behavior. And what I'll posit is not killing leukemia cells, but rather re-educating, rehabilitating them, reminding them that they can actually be normal cells and normal members of the hematopoietic hierarchy. So today I thought I would focus on one specific class of epigenetic proteins because there are many different mutations in epigenetic regulators. There are those that affect chromatin modifications, nucleosome assembly. But today I would focus on specific biochemical mechanism, and that is the conversion of unmethylated DNA, cytosine methylation, from unmethylated to methylated DNA, to hydroxymethylated DNA, and back to methylated DNA. This is a fundamental core concept of epigenetics, the idea that by methylating DNA, that the DNA methyltransferases like DNMT3A do are important in gene silencing, at least when these things occur at promoters, although that's a vast oversimplification. And we now know from a lot of work from many labs, Angela Rao, Nathaniel Hines, and many others, that the TET family of enzymes, TET1 and TET2 and TET3, convert methylated DNA to hydroxymethylated DNA, and that then subsequently leads to DNA demethylation. So it's a fairly true statement that loss of TET function will block this conversion, accumulate methylated DNA, and deplete hydroxymethylated DNA. We know from a number of labs, including our own with Craig Thompson and Ari Melnick, that IDH mutations, which are mutually exclusive with TET2 mutations, at least in leukemia, by producing an alkyl metabolite 2-hydroxyglutarate, poison TET enzymes and block this step. So we know that either loss of function mutations in TET2 or gain of function mutations in IDH have the same thing, that they block this step. And what are the implications of this? Is that you get a profound depletion in levels of hydroxymethylcytosine on the right in patients that have IDH or TET mutations. You see an increase in methylcytosine, but I'll point out that a minority of the methylcytosine ultimately is hydroxymethylated. There's a lot more methylated DNA in the mammalian genome than hydroxymethylation. And there are other carboxy and formyl that are even less substantive. So the actual obvious thing, if you're actually profiling patients, the thing that allows you to pick out the TET and IDH is the loss of HMC more than the gain in methylation, although you can actually see both of these. So we've been interested fundamentally in two questions. The first that I'll tell you about for a few minutes is an area that we've been working on for many years, which is, are these the only mutations, TET and IDH, that have this phenotype? Are there other AML or cancer mutations that can affect this pathway? The second is, once we understand which are the mutations that affect this pathway, can we understand mechanistically how they contribute to transformation? So I'll touch these one after the other. So the first question we had was, are there other mutations that have the same phenotype as TET and IDH mutations? So we went back to our 500 patients where we'd done sequencing and asked, are there other mutations that are exclusive of TET and IDH mutations other than each other? And a mutation which we wouldn't have expected to have the same phenotype rose out at us, and that was mutations in the Wilms tumor 1 gene. And I'll remind you that in AML, mutations in WT1 are clearly loss of function, nonsense, and frame shift mutations. And we found essentially no patients that had coincident WT1 mutations with IDH and TET mutations. We then looked at the DNA methylation of these WT1 mutations with Ari Melnick, and we were able to show that with the caveats about the difference between methyl C and hydroxymethyl C, the WT1 mutant patients are characterized by profound hypermethylation compared to normal bone marrow or compared to non-WT1, non-TET, non-IDH mutant leukemias. So they have the same hypermethylation phenotype that you would see in an IDH or TET mutant leukemia. And then we went on to measure hydroxymethylcytosine. As I said to you before, that IDH and TET have a reduction in HMC. Look at the WT1s. They have the same defect in hydroxymethylation that we see in IDH and TET. So this leads to two fundamental questions. Can we look not just at global levels of HMC, but actually look at site-specific changes in HMC? And second, what's the mechanism? How does WT1 affect TET2 function? So the first thing we did was work with Lucy Godley and Ari Melnick to do the first, as far as we know, genome-wide studies of hydroxymethylation in AML. There have been some studies done in normal cells by Lucy and Chuan He and others. And we took a set of AMLs with and without TET and IDH and WT1 mutations and did genome-wide HMC sequencing. And we were able to map HMC in the genome of patients that don't have WT1 or TET or IDH mutations, those that do. And you can see, for example, this gene, there's a marked, about 80% reduction in the amount of HMC compared to non-IDH, non-TET, non-WT1 mutant patients. And we could basically look for areas where you had loss of HMC compared to normal bone marrow or compared to non-mutant patients. And if you then quantify what are the differential regions, the areas of HMC that are lost, what you see is very significant overlap between the TET and the WT1, and both of these are actually a subset largely of the IDHs, and we know that IDH mutations poison very significantly all three TET enzymes, so you get a global loss of TET function versus haploinsufficiency for one of these. So we actually see a much more dramatic effect of IDH mutations than TET or WT1, and we think that that's not surprising based on the biochemistry. One interesting thing, which is a bit of a sidelight, but I think is a really important one, is the following. One of the sort of vexing challenges in the cancer epigenetics fields for the last 2 decades has been that if you do measures of DNA methylation at a gene-specific level compared to gene expression, everyone says you never see good correlations, that DNA methylation does not correlate with gene expression. So that's shown on the left. We get a R squared of about 0.3. So there's a very, very weak association. If you instead look at regions that gain and lose hydroxymethylation, the R squared is 0.7 with a p-value that's 3 logs better. So it turns out if you want to predict in leukemia, which are the regions that are going to change expression based on DNA modifications, it's differential hydroxymethylation. That's the labile mark that actually predicts the regions that have differential gene expression. And we think that's important because all of the studies done by us and others in large sets of samples are always done for methylcytosine. But we think that that's a fundamental problem. And we need to actually redo these with hydroxymethylcytosine because we think these may be the key loci that predict which genes are expressed and not. We then went on to show that WT1 loss, if we knock it down or we knock it out or we express mutations, directly reduces HMC levels, so it's a direct effect of manipulating WT1. And importantly, if we overexpress WT1, we increase HMC. We've actually gone on to develop overexpression models of AML. There are models where WT1 is an oncogene in the same tissue, in AML. And we could show that those models, WT1 is important in driving AML in concert with other genes, and those models have high HMC. So we think this is a dual oncogene and tumor suppressor that even in the same lineage can toggle HMC up and down to turn on and off different genes. And we think that's a really intriguing idea about how a single gene can actually have effects in both directions. And last but not least, we've been able to show biochemically that what WT1 does is directly bind TET2 and TET3 for that matter. And in CHIP studies, we can show that WT1, when you knock it down or knock it out, you lose the ability of TET2 to bind to some of its target loci, in fact, including key transcription factors downstream. So what we think WT1 is doing is directing TET2 to specific regions to remove HMC and turn on genes. So that leads to a question. One of the ways transcription factors turn on genes is by removing DNA methylation, and we think one mechanism it can do it is by recruiting the TET enzymes directly. And there are other groups that have now actually shown the same association. We think this is a really exciting idea. So the message from this part is that we can add a third gene to the story of methylcytosine, hydroxymethylcytosine, and that is Wilms tumor. So for the last 2/3 of my talk, I want to go from sort of the epigenetic biochemistry to hematopoietic function. I'll review some data you've already heard about just for a minute or two because it'll set the stage for some of the leukemia work I'll tell you about. Because the question we've been interested in is how do any of these mutations contribute to hematopoietic transformation? And so as you heard both from Tim and from Ben, many groups, including ours, have been interested in the question of clonal hematopoiesis as patients get older. And I won't belabor this point, but just remind you that this was an oft-debated and oft-discussed entity of unclear significance. And the original observation actually was done in the 70s when investigators in the UK, using Bert Vogelstein's recently described G6PD assay, showed that some patients who achieved molecular remission and ultimately are cured of leukemia had clonal hematopoiesis at the time of remission and cure. So this idea that these patients don't have leukemia but have clonality was there from the beginning in the 70s. This is not a new concept. And so the question fundamentally is not just does it exist, but what can we understand about it and why isn't it leukemia? Why are these mutations fundamentally not causing leukemia by themselves? What do they need? What are they missing to actually cause leukemia? We went on to show, as you heard, that there are mutations in TET2 that occur in some of these folks. I'll just point out that at least from our analyses, the mutations that occur in patients that have clonal hematopoiesis are identical to the mutations we see in leukemia. They're catalytically dead, they're nonsense, they're frame shift. We see the entire mutational spectrum. So it's not like you can say, well, these are hypomorphic alleles that don't occur in leukemia. They're exactly the same. We can find no difference in the type of mutations that occur in people that have clonal hematopoiesis from those who have overt leukemia. And so this all suggests that there's this pre-malignant clonal state that can occur in patients that don't have leukemia. I'll point out that 2 groups, Ravi Majeti and John Dick, have done similar studies where they've taken overt leukemic samples, fractionated out based on cell surface markers, a pre-leukemic clone, and shown that the same mutations that Ben and others have now shown in population studies occur in normals can be shown to occur in pre-leukemic cells that occur in parallel to the overt leukemic cell. So just like there are 1 mutant cells that occur in normal people as they get older, there are 1 mutant cells in leukemia patients that probably persist and could care less about anti-leukemic therapy. And so the fundamental question is, what does this have to do with hematopoietic function? I'd point out, and I remember this from when we submitted our TET2 paper in elderly a number of years ago, and the biggest critique about our paper when we submitted it for publication was that our lab had knocked out TET2 and showed the same phenotype, that there's a clonal outgrowth without overt leukemia. And the critique from the editors and the reviewers was, well, you had already shown it's true in mouse, so it's not surprising it's true in patients. And obviously, those of us who think about patients, there's many things that can do phenotypic changes in mouse that have never been shown to be relevant in the human. And this really, I think, is really reassuring, because all of the mouse models for all these things cause stem cell overgrowth or increased self-renewal, but do not cause overt leukemia, and they resonate really well with these population studies. So that leads, I think, a fundamental question. These mutations can initiate a clonal outgrowth. We know that they can cause a clonal hematopoiesis, but not leukemia. So what's their role in actual overt leukemia? What are TET and IDH, DNMT, and WT1 doing in overt leukemia? Are they just there at the beginning to just increase clonal outgrowth and set the stage for other mutations? That's one possibility. But there's another possibility. The other possibility is that in addition to their role to initiate self-renewal, they have a second role. That when another mutation occurs, they can actually cooperate with these other mutations to rewire the epigenome a second time at the time of transformation that is distinct. And that's important because that would imply a role in leukemia maintenance, that it's not a hit and run role, but rather a role once you actually have a leukemia, and that it's reversible even after you go over the bar of transformation. So I'm going to try and argue that that's the case in the next few minutes. And so one challenge has been that until the last 2 or 3 years has been, I would posit, the lack of really good animal models in AML. I would posit that virtually every study that's led to a drug being tested in the clinic until about 2011 was either based on a cell line injected into a mouse or based on an MLL mouse model. And those are very good mouse models, but if you can envisage that, that means in many cases that drugs get to the clinic and are tested in patients where that genotype never occurred in a model, and so you're testing drugs in the clinic, and you might as well, throw it up against the wall and see if it sticks if the patients don't have the same genotype as the model you tested. And in a disease where you have 15 to 18 distinct gene expression clusters, the heterogeneity basically you're not taking into account. And so our lab and all the other labs you heard about from today are trying to now make a spectrum of genetically engineered models, primary-derived xenograft models that recapitulate the intra- and interpatient heterogeneity in AML, because we think that drug development needs to test different drugs in different subsets so that we don't just treat all of AML as one blob, because as I showed from our first slide, that's not exactly been working out so well. And so our efforts have been to develop mouse models of AML, and I'll just tell you about one of our mouse models. This is a mouse model where we crossed the FLT3-ITD knock-in that Gary Gilliland made a number of years ago with a TET2 conditional knockout mouse. So you're expressing from the endogenous locus the FLT3-ITD with loss or haploinsufficiency for TET2. Reassuringly, these mice develop a lethal disease with big spleens. So that suggests that we had a phenotypic cooperation between these mutations. And you can see that you get a big spleen that's much more severe with these two things together than either alone, suggesting maybe there's something about these two mutations actually working together, and they're not just additive, but rather synergistic. And if you look phenotypically at these mice, they have acute myeloid leukemia. Morphologically, flow cytometrically, they die of anemia and thrombocytopenia. They have blastic involvement of their bone marrow and spleen. There's nothing about these mice that looks any different to us than a classical AML patient. There's two other really important points I'd make. The first is that haploinsufficiency for TET2 gives the same phenotype. Many patients only have monoallelic TET2 mutations in concert with FLT3-ITD, so you don't actually need to delete TET2 to get this phenotype. The mouse genetics very nicely mirrors the human genetics. And the second is that a number of groups, Scott Lowe, Pierre-Paul Pandolfi, Tak Mak, and others, have used retrovirus or transgenic approaches to express IDH mutations with FLT3, and it's the exact same phenotype. And we've worked with Craig Thompson to make a knock-in mouse for the IDH2 mutation, and we have the exact same phenotype. There's literally no difference, whether it's TET or IDH, and we now have data for WT1 that it's the same, suggesting that this is a class effect, that altering hydroxymethylation cooperates with FLT3 to develop AML. And if you look at these mice in their therapeutic response, one important thing we've noted in these mice is that they don't respond to chemotherapy. So the way we always do these experiments, all of our therapeutic studies, we engraft equal numbers of leukemic and normal cells, and then we treat the mice with 7 and 3 chemotherapy. We wait 3 weeks after nadir, when it count recovery, and we ask, is it leukemic hematopoiesis coming back or normal? And with these mice, they all come back with leukemic hematopoiesis. And the response rate, at least in our series of FLT3, TET2 leukemias, or even TET2 leukemias as a whole, is about a 25% CR rate. So these are probably among the least responsive leukemias that we've seen, other than maybe the complex karyotype p53 mutant leukemias. We think that's a very important point. So we wanted to go from the phenotypic description of this mouse to actual mechanism. And so the first question we asked was a simple one. Do TET mutations or IDH mutations augment receptor tyrosine kinase signaling by the mutant FLT3? And I can tell you that you look by Western blot here, you see activation of STATs and AKT and the FLT3 mice on the right. The activation, when you also have a concomitant mutation in TET2, is no different. So there's no augmentation of signaling. It's not like FLT3 is now a better oncogene because TET2 is deleted or haploinsufficient. You get the same cooperativity that you see with FLT3. So we took a step back to the drawing board and asked, fundamentally, why do mutations cooperate to induce AML, or you could say any other cancer? And so because I trained with Gary Gilliland, I've always been a believer in this idea of the two-hit leukemia hypothesis, that you can have 2 different mutations, one that affects oncogenic signaling, one that affects differentiation, and they each confer to a leukemia a different biologic feature that together give AML. It's a little bit like Hanahan and Weinberg's hallmarks of cancer, that different mutations can bring different sort of parts of the puzzle together, or maybe like the Reese's peanut butter cup commercial when we were kids with the peanut butter jar and the chocolate bar falling in, if those of you who remember that, or maybe that's just from my childhood. But we thought maybe that's not the right model to think about cooperativity with epigenetic genes. So our hypothesis was that it's not that they each have a distinct role that adds together to give the leukemia, but maybe there was convergence. Maybe what happens is that there are some mutations that actually can coordinately regulate the epigenome of specific targets. And there might be genes that are only dysregulated when you have the right combination. That each of them could change. One of them changes methylation a little bit. The other recruits a transcription factor. But only when you get the 2 together, bam, do you actually get the critical either activation or silencing of a key target gene. And so that was sort of our hypothesis we wanted to test. Could there be specific alterations that only occur in double mutant cells that don't occur in single mutant cells? Before we did that, we had to know what population to study. And so we took a step back and profiled these mice and looked at their stem cell populations, their progenitor cell populations. What we observed was that the TET2 knockout mice have an expanded stem cell population, about 1.5-fold. The FLT3 ITD mice, which is known, actually have a reduced stem cell pool. That's actually well described because the kinase probably exits these cells out of quiescence. And the double mice actually have the expanded stem cell pool. So TET2 loss augments or restores the stem cell expansion that is not present in FLT3 ITD. By contrast, the FLT3 mice have an expanded progenitor pool. That's true in the double mutant, but the TET2 mice don't. So that means you have 2 pools that are expanded in these mouse, the stem cell pool and the progenitor pool. So we wanted to ask, where do we profile? And so we did 2, I think, really simple experiments. The first was we transplanted these into secondary recipients and showed that only the stem cell pool could initiate leukemia in secondary recipients. And I have to say, at the forefront, I'm not a big believer in leukemia stem cells or cancer stem cells, but the data is the data. We could define a specific small subpopulation in this mouse model that could repopulate into second recipients. And I can tell you, we've done similar fractionation studies in humans, and these same populations are present and abnormal in humans with FLT3 and TET2 mutations. This same LSK is actually more of a multipotent progenitor, not a true long-term hematopoietic stem cell. So that wasn't enough. I told my postdoc, Alan Shee, that he couldn't just show us what a stem cell was. He had to show it was therapeutically relevant, because if it wasn't therapeutically relevant, it's not worth studying in detail. And so he decided the best thing to do was to take the most exciting drug in this type of leukemia, which are second-generation FLT3 inhibitors, which have been shown in the clinic to have complete responses, but they're always transient. We give these patients AAC220 and other drugs. They develop complete responses, and they all come back at 12 weeks with resistance, either genetic or resistance mechanism not otherwise specified. And what he showed was nice target inhibition. We get very nice reduction in signaling. We get nice reductions in white counts and in spleen weights. But what we get is no reduction in stem cells. And if anything, we get an expansion of stem cells, and we enhance propagation into secondary recipients by treating the primary recipient, suggesting that the progenitors, which are the bulk of the leukemia, are exquisitely sensitive to tyrosine kinase inhibitors, but the stem cells are not. And so we thought this was good because people talk about stem cells being resistant to therapies a lot, but we at least were able to show in this model that it was true in vivo with secondary transplants. So then I said, all right, now you can profile the stem cells and ask, are there things in the circuitry of these stem cells that these two mutations together cooperate to induce? And he did, and I'll just skip the summary slide and cut to the chase. And what we showed is that the AML stem cells on the left had a very distinct gene expression profile from the progenitors, and they actually were more similar to normal stem cells. So they actually look more like the cell from which they came, from a normal mouse, than like the leukemic progenitors. So there are very significant differences in their expression. But there were two things that we observed that were quite dramatic. The first was an increase in ES cell or MYC gene expression signatures. And because Jay Bradner's drug has been tried in every other cancer model I can think of, we've decided not to work on JQ1 in this model. And rather, we decided to work on the other. And that was the interesting observation that the GATA transcriptional effects of GATA1 and GATA2 were totally wiped out in this leukemia. And to us, that was interesting because we were looking for silencing of a program, loss of a cell identity program. And so we thought this idea that GATAs were silenced would be something to follow up on. We, in fact, showed that the silencing of GATA1 and GATA2 is only true in the stem cells, and it's only true in double mutant cells, TET2 and FLT3. If I show you TET2 only or FLT3 only, GATA1 and GATA2 levels are 20% reduced. So GATA1 and GATA2 are only silenced, and they're only silenced in the stem cells with both mutations. So the classic definition of epigenetics, genetic context and cell compartment matter, and that's how these genes are silenced. And this is actually very consistent with Pierre Paulo's data that in the FLT3 IDH mice, he sees silencing of the GATA signature. So we think that this is a general theme of this subflavor of leukemia. So we wanted to go from gene expression to DNA methylation because we thought that might give us a hint into that. And so we did DNA methylation of the purified stem cells from normal mice, from FLT3 mutant mice, from TET2 mutant mice, and the doubles. And we said, if we're right, what we're looking for are genes that are only methylated or silenced or lose HMC, we think proximally, in the double mutant mice and not true in the single mutant mice. To our surprise, it was incredibly illustrative. What we observe is an explosion in the number of genes that are hyper and hypomethylated in the double mutant cells. And it's a vast excess compared to TET2 only or FLT3 only. Yes, TET2 only mice have hypermethylation. That's been described by us and others. But the hypermethylation in the double is 2 logs greater. So the effects of on methylation require this oncogenic synergy between these two genes. And then we took all of the genes in the genome and we ranked them from 1 to 20,000. We said our criteria are genes that are only methylated with the 2 hits together and have a change in gene expression. Seemed like how we define cooperative target genes. And number 3 on our list was GATA2. And that was obviously reassuring to us. And there's actually some pretty interesting other genes on that list, like HOXA3 and MN1 and HOXB3. So we're not saying that GATA2 is the only answer, but the fact that we could detect GATA2 in an agnostic fashion at the top of our list was quite intriguing. And if you look at the DNA methylation peaks, we see a dense methylated region only in the TET2 FLT3 and not in the others. So this is unmethylated all the way to densely methylated, suggesting GATA2 is only methylated in the double mutant stem cells and not in the single mutant cells. All right, so that's sort of where we were. We wanted to ultimately go on and move on, and I'm just going to skip this point and get to the money experiment. So I said to Alan, you got to do one more. You got to put back in GATA1 and GATA2 and see what's happened to the leukemia. Because if these are the key targets, the leukemias should change their behavior. Maybe they'll stop growing, maybe they'll change their cell identity, maybe they'll differentiate, or maybe nothing will happen. And his response, of course, to me, because he's a really careful guy, is that you're telling me I need to take one of the 10,000 targets and get a phenotypic change. And I said, you should do the experiment. And what he observed was complete disappearance of leukemia by adding either GATA1 and GATA2. The fascinating thing was that we could track these cells for 6 weeks in vivo, and they actually go in, they home, they engraft, and then they differentiate and go away. So the cells are not apoptosing. They're actually undergoing normal hematopoietic differentiation and then going away. So it's really what we believe is that these are epigenetically silencing a normal developmental pathway that these leukemias are trying to repress, and by adding back ADH2, you're releasing that block. And you're reminding the cell that there's a way back to normalcy, just like somebody we want to rehabilitate. This idea that we can add back GATA1 and GATA2, as Craig Thompson likes to say at our place, we've spent 30 years trying to teach cancer cells to die and not done so well. So maybe it's time to rethink this and re-educate the cancer cell, rehabilitate it, and redifferentiate them, and not just expect them to commit suicide. So this really suggests that there's a very distinct role of TET mutations, or IDH, or WT1, in addition to their effect on self-renewal, that they can actually cooperate with other mutations to get this synergism, to epigenetically regulate key target loci. And that's only true in the overt leukemias, not in these early precursor states. And we think that TET2 is sort of like a disaster waiting to happen for a second hit to occur. And when it does, it's carried along for the ride and it rewires the epigenome in a second way. And we think that's really important. And the important thing, I think, of the GATA experiment for us was the proof of concept that this is a reversible phenomenon. These cells are not irrevocably leukemic. They can be restored to a normal phenotypic state that can't no longer outcompete cells. So we've wanted to now go on and therapeutically test that. And that is a non-trivial enterprise. The challenge with TET-mutant leukemias is that we don't have drugs that can reactivate TETs. We do have hypomethylating agents, and I'll show you a slide or two with them in a minute. But obviously the low-hanging fruit here is that IDH gain-of-function mutations, I said before, inactivate TET. And so there's the appealing idea that you can pharmacologically block the neomorphic IDH function and ask what its effect is on the leukemia. And so this is a little bit of our data, which is very preliminary, with 5-azacitidine in this model. We treat the mice for 9 weeks. So if you look on the left, with azacitidine alone, or by adding azacitidine and a FLT3 inhibitor, we actually get a precipitous decline in the number of blasts, in the number of white cells, and in the spleen size. We get a complete response pathologically with azacitidine in the TET-mutant leukemias alone or with a FLT3 inhibitor. The interesting thing, though, is that if you ask what proportion of the hematopoiesis in these mice is mutant, because these are competitively transplanted with normal cells, so we can look at the relative effect, all of the hematopoiesis in these mice is leukemic. So what happens in these mice is they restore their normal red cell and platelet development. They're no longer anemic or thrombocytopenic, but it's coming from the leukemic clone. And if you stop the azacitidine, the leukemias come back. So you're not actually debulking of leukemia. It's a constant redifferentiation signal. Maybe if we treat for a year in a mouse, we could get to the point where they actually would go away. But what you're getting is a constant redifferentiation. However, and these are early data from just a couple weeks of therapy, if we add a FLT3 inhibitor, we start to see a reduction in leukemic burden. So we think the answer will be combining our differentiation therapy with cytotoxics, with TKIs. And the critical question is that Steve Balin and JP Isso were right. It's going to be sequence and dose. You can't just add them. And I think we need to be thoughtful about how we do these studies. It's just like combining epigenetic therapies with immunotherapy. You can't just add the two drugs and hope for the best. In fact, we would argue if you add them together at the beginning, you might actually precipitate toxicity and myelosuppression. You need to differentiate the leukemia cells and then come in, for example, with the tyrosine kinase inhibitor. But we wanted to move from hypomethylating agents, which are at best a crude class of agents, to something more specific. And so we've worked with our colleagues at Agios to help them develop one of the first drugs in a novel class of IDH inhibitors. And many of you are probably aware that they've been developing IDH inhibitors for both the IDH2 and IDH1 mutations. So we've been doing a lot of that work with them, initially using FLT3 ITD IDH2 mutant mouse. And you could see, and this is the critical experiment, that they completely aggregate serial replating in vitro. So you block stemness or self-renewal of these cells. They actually differentiate in vitro if you treat them with an IDH inhibitor, and it's very concomitant with target inhibition. We've gone on to treat both mouse models, and we get very nice reductions in the proportion of 2-HG, which is the metabolite made up of the IDH mutations. We get a nice reduction in the proportion of blasts. These are in genetically engineered mouse models. If we do this in PDXs, we get the exact same answer. And so we thought this was encouraging preclinical data. They did a lot of PDX work in collaboration with investigators in Paris. And this really led to the stage of the first-in-man IDH inhibitor trials, which are being done at both the MGH and the Dana-Farber and at Sloan Kettering and a number of other places. And one of my colleagues, Eitan Stein, is working with a number of investigators to lead these efforts. We know and this I think is really exciting, is that even at the first and second dose cohort, there was potent target inhibition. I think this was one of the really exciting or fortunate things about how fast this has moved, because even at the first 2 doses in a phase 1 trial, you're getting 90% target inhibition. That doesn't happen very often, and I think that's because the drug had very little toxicity in preclinical studies, so they started at a very high biologically active dose. But obviously the proof is in the therapy. And we now know that about 30–40% of the patients on the phase 1 trial have achieved either a complete response or a complete response with incomplete platelet recovery. And this is primary refractory, relapse refractory, post-transplant AML, patients who have no options. So these are the worst of the worst. And in a drug which essentially has no toxicity, we see profound responses in a subset of patients. We need to understand why it's only some and not others. We need to understand, will it be maintained and curative? And probably most excitingly, how do we combine this drug in a way with other drugs to actually get this thing up front to our patients at a point where we can cure people before they relapse? And so we think this is encouraging, but it's just the beginning of what we think will be a very long story, and obviously not just relevant for leukemia, but for all IDH mutant cancers. And we hope if we can make better epigenetic drugs for other kinds of cancers that have TET or WT1 or name your favorite epigenetic protein. So in summary, specific combinations of mutations, including FLT3 and genes in the TET2 pathway, co-occur in AML and are associated with bad outcome. We know that mutations in epigenetic modifiers increase self-renewal and increase predisposition to AML. They're initiating hits. But they have a very different role in overt leukemia, where they get co-opted by other oncogenes and tumor suppressors to rewire the epigenome at the time where the leukemia develops, overt leukemic transformation. But I think the critical thing is that none of this is beyond the point of no return. We can reprogram and redifferentiate these cells even after other mutations occur, really suggesting that there are ways to approach cancer, I think, that are distinct from the way maybe we thought about it 20 years ago. And it's our hope that with approaches like this, combined with all the other exciting ideas, immunotherapy, transplantation, cellular therapy, that we can ultimately cure a lot more of our leukemia patients and obviously other cancer patients. I'll just acknowledge the folks that did all the work. Alan has led all of the work on TET and IDH in my lab with Caitlin and Brittany. Rajit was the one who figured out what WT1 did, working with Ken and Ari and many, many others. We've had lots of collaborators at Sloan Kettering, particularly Craig and his lab and Eitan on the clinical front. And we're very grateful to our collaborators at Agios as well as Lambert Busk for the clonality work. Thank you very much for your attention.
I just think it's great that you're standing in a former jail talking about rehabilitating leukemia instead of incredible. There were questions out there? Yep.
So I'm actually fascinated with the, thank you, I'm fascinated with the data you presented on GATA1 and GATA2. Looking in the double mutant, if you look at the MDS and AML as a continuum, one of the things you see in MDS, megakaryopoiesis is not necessarily normal. You can see erythropoiesis, not normal. GATA1 and GATA2 both drive or are responsible for erythropoiesis, megakaryopoiesis. Without them, that gets screwed up. Then you showed 5-azacitidine, which has been around for a while. That got me thinking as well. I have a PhD, not an MD. Looking at GATA1, if you put that into cells, you can actually open up the erythropoietin receptor. You can open up the alpha globin and beta globin, the entire gene loci, just with GATA1 and GATA2. I'm wondering if you've looked at any other chromatin aberrations that may have been repaired when you introduced GATA1 and 2.
So we haven't done what I would call the sort of distal sort of work of like what happens after you add GATA1 and GATA2. What I can tell you is that it's really interesting, and that's an area that we're going to explore. But there's a couple of really interesting things. The first is that inactivating GATA2 mutations occur in AML, and they never occur with TET or IDH mutations. They're rare, but at least we've never seen one co-occur, suggesting that if you're going to inactivate GATA2, you don't need to do it in these leukemias. The second is the HMC profiling I showed you. The loci that are most affected by HMC, differential loss of HMC, are GATA binding sites. We actually think in addition to GATA itself being silenced, that there's a TET-GATA loop where it actually feeds forward and feeds back. And the third is that Lucy Godley recently showed a paper in Cell Reports that TET loss alone affects erythroid differentiation. So we think it's actually a very complicated, intricate loop, like what Bertie Gotkins has argued about these TFs, that we think the transcription factors use differential hydroxymethylcytosine to turn on and off genes. Bringing chromatin into that will be the next thing. But we think it's a really interesting area to probe in more detail.
Other questions out there? Let me just ask one quick one. I thought it was really cool. You're showing that there's a better correlation between 5-hydroxymethyl and gene expression profiling. As you know, in the clinical literature, it's been very frustrating. Can't connect the dots between methylation, gene expression, and clinical responses to methyltransferase inhibitors. So it's an
the correlation is profoundly different. I will tell you that we did a lot of modeling with the statisticians and asked which predicts better, HMC wins, and then we tried to say to them, well, maybe if we add both. And we couldn't even show that once you account for the differential HMC, you could get an incremental that there are some additional loci where only methylation changes. We think that a lot of the labile changes in myeloid cells are the HMC. And the other areas may be just methylated background that doesn't change much or it isn't matter to the phenotype. That the labile changes that are relevant to the reprogramming, that may be simplistic, but that's the hypothesis that we're drilling down on now.
So have you had a chance to look at clinical samples to see, responders, non-responders, whether?
We haven't done that yet, and that I think is something that maybe we should do. What we're actually going to do next is look at the azacitidine responder mouse and see if HMC changes. But I think there's a lot of interesting things to do there. Bruce?
One question. So does the regulation of gene expression stop with just hydroxylation of the methyl group, or do you have to actually remove hydroxymethyl?
Oh, that's a complicated question, Bruce. We know that the rate-limiting step, at least in a test tube, is the conversion of methylated to hydroxymethyl, and if you leave that cell alone, it will unmethylate. There's a huge debate, depending on the cell type, whether the subsequent steps, which can include carboxyl or formal, are required, rate-limiting, cell-specific. There's been a lot of data that the AID and APOBEC pathways can be important there. This is, I think, an area that the biochemistry has all been done in 293 T cells, and we need to actually get into cancer and normal cells and do, because I bet it's going to be very cell type specific.
That was a great talk, as always. What are your thoughts on how you get from FLT3-ITD to GATA1 or MN1? Do you think it's an actual transcription factor downstream of those, or a phosphorylation change in epigenetic?
That, to me, is the most interesting part. So we know that in the TET2 only mice, there's almost no change in GATA expression. In the FLT3-ITD only mice, there's about a 40% reduction in GATA1 and GATA2 expression. Moreover, if we treat these mice with a FLT3 inhibitor, you bring it up about 30%, but not all the way back up. So I actually think FLT3 is the initiating event that's recruiting a repressor, and then the TET loss actually takes a modestly silenced gene to a fully silenced gene. And that's an area that we're starting to look at, and like, what are the motifs? Because you would have thought of being a transcriptional activator. When we've done cell line studies and mouse studies with chronic FLT3 inhibitor treatment followed by RNA-seq, 90% of the genes go down, not up. And how much of that's feedback and what Neil Rosen would talk about, I don't know. But we see in a chronic therapy, not with the 3-hour, if you do chronic therapy in these same populations, what are the steady state changes? Most of them are repressed genes. And so I think that's an interesting area that we need to probe.
Great. Any last questions? If not, I want to thank again both of our guests.